Hello, welcome to Brain Science. In this episode of Brain Science, I want to talk about new information that's come out recently about Alzheimer's disease. Now, we all know what Alzheimer's disease is. In this disorder, older people lose the abilities to make new memories and gradually forget where their houses are located, where their car keys are, and even the names and faces of the people that are dear to them. So this is a very severe disorder. And what we all want to know is what causes this disorder and what can be done about it. New research over the last 5 years has provided some hope that there might be some treatments for Alzheimer's disease. But before we go into some of this new research, we have to understand a little bit about the anatomy of the brain. So here's a picture of the brain. In this diagram of the brain, we can see the anterior parts of the brain that are located just underneath your forehead. And on the other side of the picture, we can see the posterior parts of the brain that are located just beneath the back of your skull. Now, in order to understand what parts of the brain are most affected in Alzheimer's disease, we have to make a cross-section of this picture, flip it over, and see the brain in a little more detail. So let's do that. Here's a cross-section of the brain, and what you can see immediately is that the outside of the brain is covered by a thin layer of nerve cells, which is called the cortex. A specific part of the cortex, called the temporal lobe, is the area of the brain that's earliest affected in Alzheimer's disease and which suffers the most damage. Let's take a closer look at this temporal lobe of the brain. Here we have a cross-section of an actual brain, and we can see two major components of it. The outermost part of this temporal lobe is an area of the cortex called the lateral entorhinal cortex. Nerve cells in this part of the brain project to many parts of the brain, but particularly nerve cells project to another structure deep to it, which is called the hippocampus. And the hippocampus is a complex arrangement of nerve cells organized into several layers. And one of the layers is a particularly dark staining layer of nerve cells called the dentate gyrus of the hippocampus. In Alzheimer's disease, the nerve cells that first die are found in the entorhinal cortex. And later on, nerve cell death increases and expands to another area, the dentate gyrus of the hippocampus. Now, since the hippocampus has been known to be very important for the formation of memories, if you have nerve cells dying in this region, it's not surprising that people with Alzheimer's disease first seem to have defects in memory. Now, if you look at this area of the hippocampus more closely, you can see the very complex patterns in which nerve cells are organized. If you look carefully at one of these regions, you can see the nerve cells themselves. Each nerve cell is a little triangular-shaped cell with a round, pale staining nucleus and a little dot in the center of the nucleus. Now, millions of these nerve cells can die during Alzheimer's disease, so it's not surprising that defects in brain function start to occur. But one of the things that people haven't understood for years is, why do these nerve cells start to die? Now, during the early history of Alzheimer's disease research, only a few things were known about the Alzheimer's brain. One of the prominent features of an Alzheimer's disease brain is that we can find large amounts of extracellular material called amyloid plaques. These are little circumscribed areas of some sort of goo that accumulate in the Alzheimer's brain. Another feature of the Alzheimer's brain is that dying nerve cells acquire masses of abnormal microtubules, which are called neurofibrillary tangles, and these are seen to be some sort of a sign that these nerve cells are in trouble. But beyond this, early researchers didn't really understand anything about what causes the problems in Alzheimer's disease. However, in the 1980s, new information began to be acquired. For one thing, the protein that formed these amyloid plaques was identified, and it was named amyloid precursor protein. Now, once this protein was understood, people could try to understand more about Alzheimer's disease. And one of the interesting findings is that in about 1% of the people with Alzheimer's disease, inherited abnormalities in this protein are found. Also, if you create rats and mice that are genetically modified to produce this abnormal amyloid precursor protein, these rats and mice also develop amyloid plaques and Alzheimer's disease. So this was very heartening information, and this information led to the hypothesis expressed by John Hardy and David Alsop in 1991 that not only the inherited forms of Alzheimer's disease could be caused by this protein, but also this protein might somehow be involved in the creation of Alzheimer's disease in the remaining 99% of patients. If you want to look at some of these papers, you can go to a website called www.pubmed.gov, and if you type in the name John Hardy, 1991, this particular paper will come up and you can take a look at it. There's still some problems with the amyloid hypothesis. One of the problems is that some older people develop numerous amyloid plaques in their brains, but they never have any symptoms. They don't develop memory loss or cognitive or any other problems. So it's more recently thought that perhaps small fragments of the amyloid precursor protein are the culprits and maybe have some sort of toxic effect on neurons, and then also can accumulate into plaques, which are not necessarily themselves the main problem. However, even this type of thinking has been challenged recently because a number of clinical trials have been created that have attempted to reduce the amyloid burden in the brain. In these clinical trials, various types of medicines were given to Alzheimer's patients, and they did reduce the amount of amyloid in the brain, but they didn't cause any reduction in the symptoms of the disease. Also, a recent paper by Chris Maroney et al. In 2020 showed that if you reduce the amyloid burden in experimental animals like rats and mice, amyloid plaques diminish, but these animals still have abnormalities in memories and in cognitive function. So what is to be done if reducing the amyloid burden in these animals doesn't really have a major effect? What can be done about this disease? Well, one of the things that might be helpful in understanding Alzheimer's disease is what is so special about the entorhinal cortex neurons that die in the very first place. We know that the entorhinal cortex does not contain elevated levels of amyloid. What other special features of the entorhinal cortex are known? Well, one thing that's surprising about these nerve cells is that they send nerve fibers that connect to newborn neurons in the dentate gyrus. Now, newborn neurons is a new concept in itself. For many years, it was thought that nerve cells in the adult brain are never renewed. If they die, they die, and no nerve cells are born to replace them. But as it turns out, that's not precisely true. In the hippocampus and in a very few other areas of the brain, new neurons are actually generated and are born. For example, in the hippocampus of the rat, you can identify newly born neurons by staining for specific proteins that are only found in these cells. So here, if you look at this section through the dentate gyrus of a rat brain, you can identify dark staining cells that are actually newly born. So perhaps there's something toxic about this process of neurogenesis that affects these axons that project from the entorhinal cortex. But what could be toxic about newborn neurons? Why would they have any bad effect? Well, these newborn neurons in the hippocampus can be identified by special staining procedures. And here in this particular picture, they look like little black circles. Now, one of the curious things about these newborn neurons is that they're in direct contact with so-called stem cells. These stem cells can be identified by another staining procedure which stains them brown. So here they are right there. One possibility is that toxic influences of this process of neurogenesis can somehow affect the axons that are terminating this region, and the axons die back and cause the death of the entorhinal cortex neurons. So an understanding of this process of neurogenesis may be crucial for arriving at some sort of treatment for Alzheimer's disease. A number of features of these stem cells may have some toxic influences. These stem cells stain for a protein called fatty acid binding protein, and this protein can regulate amounts of fatty acid called palmitic acid in the environment of the axons. Now, we know that palmitic acid can enhance toxic effects of amyloid, so perhaps the combination of amyloid and palmitic acid could be leading to the death of these axons. Another feature of these stem cells found in this area is that they have a very specialized glucose metabolism. And by regulating the amount of glucose in the area, they could perhaps change the amount of glucose that attaches to amyloid. And it's known that glucose enhances the toxic effects of amyloid precursor protein, and therefore could stimulate the death of neuronal structures. There's another strange feature of these stem cells that is found in the hippocampus. These stem cells undergo an age-linked mitochondrial degeneration. So cytoplasmic components of these cells start to die off. And when they die off, They leave behind dark staining granules that are next to the nuclei of these stem cells. These cytoplasmic granules are enriched in copper, and this arrow points to these granules right here. Copper can also enhance toxic effects of amyloid. So this is yet another substance released from these stem cells that could be enhancing toxic effects of amyloid and could cause the death of neurons. Well, what can we do about all this? One thing that we could perhaps do is we could enhance the rates of neurogenesis in the hippocampus. This would cause the transformation of the stem cells from their immature forms into the mature forms of neurons, and therefore they would acquire brand new properties and wouldn't have all these toxic effects. It's already known that stimulating rates of neurogenesis in experimental animals can reduce the symptoms of Alzheimer's disease, and this possibly may be due to a compensation for the deaths of damaged neurons. Also, increasing rates of neurogenesis may deplete the numbers of stem cells, and this could reduce their harmful influence. Now, how do we go about stimulating neurogenesis? Well, physical activity or exercise can somehow stimulate neurogenesis in the dentate gyrus, and there's some information suggesting that this could be helpful in reducing the severity of Alzheimer's disease. Also, there's a recently discovered chemical called algernon that can dramatically increase neurogenesis in mice. So this possibly could have a helpful effect upon the course of Alzheimer's disease. In short, it could be possible that some of the symptoms of Alzheimer's disease could be reduced by stimulating neurogenesis by a variety of different means. If you want to look at some more information about the brain, I've written a book about it that might interest you. It talks about some of these stem cells and other aspects of neuroscience. And if you want to continue to look at some more of my videos, I invite you to subscribe to my channel and look at some more of my videos. So thanks for watching this video.